Okay, good weekend, and this is our last week of lectures, I'm sure you know. This Friday, which would be our normal lecture slot, instead of a lecture, we're going to have a review session for the course, and it'll be webcast. I did work that out with the folks here, so please do show up if you can at 8, and I'll try to review the material that I think is most important for the class during that hour, and we'll post that material on vSpace as well. Not to say that that's all you have to study, but I'm going to try to at least encapsulate the things that I think are really key concepts during that part of during that Friday session. And then that evening, of course, is the midterm. I realize the review session is the same day as the midterm, but it's the only way we can schedule that. This room's really difficult to schedule. And to get a webcast out, that's about the best we can do. And the midterm's going to include all the material up to slide 8 today. So just not including slide 8, so everything up to water transport. And then the final will be on December 10th at 7 p.m., which will be all the material with an emphasis on the later lectures, especially this week. But fair game for the this whole part of the semester, this plant fungal diversity part of the course. And of course, that will also cover Dr. Hosenbeck and Dr. Chabel's material too. Any questions about that? I won't be holding regular office hours next week, but if you have questions, write me, and also we could arrange to meet if that works out best. But I'll be gone in the middle of the week. I have to be out of town right then, so I can't meet at that time. Earlier or later in the week would work out. All right, so today we're going to move on to looking at how substances move around inside of plants, and first looking at absorption of water and minerals in the roots and ways plants avoid contaminating the vascular tissue by soil solution moving into that. And then we'll look at how sap is transported through the plant, both in the xylem and the phloem, which is really fascinating and pretty amazing in terms of the physics involved. And then we'll look at how plants can actually perceive light, which is really critical for their survival. All right, so when we think about substances moving into the roots, plants need to have the ability to absorb water and minerals freely from the roots, but at the same time, they need to be able to exclude substances in the soil that can contaminate their tissues. And this is a real challenge, and the anatomy and physiology of roots is such that there's a great solution to this problem. So these are some cells arrayed here from the epidermis here on the left toward the center of the root on the right, running radially into the root. And substances that actually pass through the plasma membrane, the cell membrane, can freely move throughout the root tissue, even into the vascular cylinder. But anything that crosses the cell membrane is subjected at that point to discrimination on the part of the plant. The plant basically controls what comes in through the cell membrane very strictly. But once things are inside the cytoplasm, inside a cell membrane shown here in orange, once it does manage to cross through the cell wall and the cell membrane, it can actually move between cells freely through the plasmodesmata, which are these channels in the cell wall that cytoplasm can move through in plants. So that's a free, once they're through the cell membrane, they can move through what's called the symplast, or basically through the cytoplasm between cells, no problem. But also, water and minerals from the soil solution can move along the cell walls and in between cells in the intercellular spaces. And this is called apoplastic movement. And the plant can't discriminate, or basically can't limit that sort of flow. There's no discriminating ability on the part of the plant. So when water and minerals come in, for example, through a root hair on the outside of the root, where they can freely move in, there's no cuticle there to prevent it. Basically, soil solution, just dissolved soil substances, can move along that cell wall, in the cell wall, in between cell walls of cells. And they can do that all the way to the innermost layer of the cortex. So this is the vascular cylinder to the right here. But this cell represents a cell of the endodermis, the innermost layer of cortex. It's just to the outside of the vascular cylinder of a root. And these cells have a strip that runs all the way around them, called the Casparian strip. It's basically just a belt of suberized cell wall. And suberin, the substance that is in the cell wall around this strip, is the same substance that's in the cell walls of cork cells in the periderm that prevents water loss. So it's a waxy substance that is impregnable to water. And so water and dissolved minerals that get to this point, if they were to continue, they would contaminate the xylem. I mean, you'd have all kinds of dissolved soil substances getting into the conducting system of the plant and fouling it up. But this suberized layer, this Casparian strip, named for the discoverer of this strip, whose name was Casper, prevents that from happening. And at that point, the only way into the vascular cylinder for substances that come along through this apoplastic route is to cross the cell membrane, at which point the plant can basically limit what comes in. At the same time, this Casparian strip is important because the plant in the roots is actively concentrating some minerals through active transport, actually using energy to concentrate minerals that are needed at a higher concentration than they exist in the soil solution. And if it wasn't for the strip, those minerals would just diffuse back out into the soil solution through the cortex and epidermis. So it holds things in and it keeps things out. And here again, we see a cell of the endodermis. This is looking down at it from the top, the transverse wall wrapping around. And if we looked at it head on from the outside of the plant towards the center of the root, we'd see that it goes all the way around this cell like a belt, as I mentioned. And of course, the endodermis is a continuous layer of cells that wraps around the root. It's the innermost layer of the cortex. And the Casparian strips of adjacent cells there's no seam there that water minerals can work through. And if you look at it again from the outside of the root facing in, it would just be this wall of suberin preventing water and minerals from coming through. So it's a really effective barrier between the cortex and the vascular cylinder of the root. And this is a little difficult to see from the back, no doubt, but you can see here at root hair, the apoplastic route again, we have movement along the cell wall or in between the cell walls of adjacent cells. But at this point, this apoplastic route shown in purple is no longer available to go further to the interior. You have to actually cross the cell membrane to get through the endodermis and then into the vascular cylinder. Whereas anything that managed to cross through the cell membrane, for example, here in a root hair, has a free route all the way to the vascular cylinder. Okay, so that's the function of the endodermis, basically to prevent contamination of the vascular cylinder, prevent minerals that have been concentrated in the vascular cylinder from leaking out. So what happens how is it then that water actually gets through the whole plant? This is an amazing type of mechanism that allows a plant to basically move about a ton of water a day. An average sized tree can move about a ton of water a day from its roots up into the leaves without any energy expenditure by the plant at all. So biology and evolution have really overcome physics in the case of water transport in plants. And before talking exactly about how that happens, it's important to understand the different kinds of mechanisms that allow substances to move within plants. One of these is just diffusion, simple diffusion, which is passive transport. That's a slow process, and it's far too slow to allow water to move through plants to the extent that it needs to. But it does explain a lot of what's going on between individual cells that are adjacent to one another. And that's basically just diffusion across a membrane, cell membrane, spontaneous movement of a substance down its concentration gradient from higher concentration to lower concentration. That's not an energy requiring process, it's just simple physics. Active transport, on the other hand, is movement of a dissolved substance. We're not talking about water in this case, but some dissolved substance, solute is what we call a dissolved substance, across a membrane against its concentration gradient, from going from lower concentration to higher concentration. And that requires an energy expenditure on the part of the plant to accumulate something at a higher concentration than it's present in the ambient environment. Okay, so that's active transport. That also is happening across short distances. And then bulk flow is basically the most important type of mechanism we're going to talk about today, that is basically the movement of a fluid due to a difference in pressure between two locations. But these other types of transport are going to be important too to keep in mind, as we'll see. All right, so when we look at, let's just talk about movement of water between adjacent cells. And basically movement of water across a semi-permeable membrane is what's called osmosis. And this basically demonstrates osmosis in this slide, where we have a U-shaped tube here open at the top with water in it, and we have dissolved sugar at a higher concentration on the right side here than on the left side. And these little green dots are sugar molecules. And here you see a semi-permeable membrane separating the two halves of this U-shaped tube. So this membrane is permeable to water, but it's not permeable to sugar molecules. So we call it a semi-permeable membrane. And water molecules, of course, are quite tiny compared to these dissolved substances. The dissolved sugar is shown here in green. The water is, you can see the oxygen in red and the two hydrogen atoms in white here. And basically the point here is that with a higher concentration of sugar molecules to the right side, water is going to bind to those sugar molecules. And we're going to have fewer water molecules that are free to move on the right side of this membrane, because more of them are going to be bound up with sugar. And so just through simple diffusion, water is going to be moving more often to the right than to the left, because there's a higher concentration of free water molecules on the left than on the right. And eventually we'll come to an equilibrium with equivalent concentration of water on both sides of that U-shaped tube. All right, so in thinking about how things actually get through membranes, simple diffusion can account for hydrophobic molecules getting across this hydrophobic membrane. It can also account for water molecules. Water molecules are obviously charged, but they're so small that they can actually diffuse across the membrane. But they actually move across the membrane more quickly than simple diffusion would allow for. And they're moving at this more rapid rate because of facilitated diffusion. There are proteins embedded in the membranes that allow for charged molecules to move more quickly through the membrane, still without requiring energy from regions of higher concentration to lower concentration, but at a faster rate than through these channel proteins like you see here, or through proteins, these carrier proteins that change conformation after they bind a molecule on one side and then are released on the other side. So basically there are proteins that help water and other molecules get across membranes, charged molecules get across this hydrophobic membrane without just going down the concentration gradient. So again, without the use of energy, this is still passive transport. Active transport is going against the concentration gradient. That requires the use of energy on the part of the plant and also involves proteins. And we're not going to get into that too great detail. We'll talk about it again a little bit more in terms of active transport with regard to the functioning of stomata in a moment. So the direction of water movement within a plant is going to be dictated by two things: the concentration of solutes within different parts of the plant, and also physical pressure. These are the two things that are going to cause movement. So let's think about these with an example of some cells in solution. So basically the osmotic equilibrium between a cell and its external environment is going to be partly dependent on the relative solute concentration, the concentration of dissolved substances inside the cell versus outside the cell. Okay? And so here we see a typical case for a plant cell where the solute concentration of dissolved substances are at a higher concentration inside the cell than they are to the outside. And so just as we talked about, osmosis is going to result in net movement of water to the inside. Water is freer to move in this direction than in this direction, in rather than out. Water is more bound up with solutes on the inside than outside. And so net movement in. And thing to consider, plant cells have this cell wall around their cell membranes. It's pretty rigid. And that cell wall is going to push back against expansion. If the cell continues to expand against that cell wall, the wall, which is fairly rigid due to its makeup, cellulose, is going to resist that further cellular expansion. And that will exert a positive pressure that results in osmotic equilibrium, even though there's a higher solute concentration inside the cell than there is outside. So both the solute concentration, as I mentioned, as well as the pressure are important with regard to movement of water. Now in this case, there's the solute concentration inside the cell is the same as outside. And so the cellular contents here are not under pressure at all. Water is just as free to move out as it is to move in. And the cell is not going to be turgid. Turgid is what we refer to if the cell is under positive pressure. And turgidity is what, in part, maintains the rigidness of the plant body, particularly the parts that don't have very much structural support, like leaves or young stems. And so under this condition, we're going to have wilting of the plant. The cells are not going to be turgid. The parts of the plant that are not well structurally supported by lignin are going to, or parenchyma, for example, are going to wilt. And if the solute concentration inside the cell is actually lower than outside the cell, then the cell membrane will actually pull away from the cell wall, and the whole cell will collapse, and then it's all over for the cell. It's plasmalized, which is a seriously unhealthy condition. But it is possible to recover from wilting, as you know, if you've had a wilted plant and watered it up to a point. All right, so in order to think about water relations in plants, it's really important to think about this concept of water potential. And water potential is basically just the potential energy of water And it's basically the freedom of water to move within the plant. I mean, it applies to more than just plants, but in terms of the plant environment, it's the freedom of water to move. And basically free water moves from regions of higher water potential to regions of lower water potential, which makes perfect sense given osmosis. If water potentials are negative, then higher is less negative, and oftentimes water potentials are negative. So you need to think about whether we're dealing, for example, in this case, negative 0.7 megapascals. Water potential is measured in a unit called megapascals. Whether that is higher or lower than negative 0.9, of course it's higher. The symbol for water potential is the Greek symbol psi shown here. And there are two components to water potential, as we just mentioned. There's solute concentration and physical pressure. So those are the two things that control water movement: solute concentration, physical pressure. The higher the solute concentration, the lower the water potential. The less free the water is to move. It's bound up with solutes to a greater extent. And the higher the pressure on the solution, the freer the water is to move. Obviously, if you put water under pressure, it's going to move, and that means higher water potential. So let's look at an example here with the cell in the center here. So there are two components: the pressure component of water potential with this subscript P, and the solute component of water potential with subscript S that are additive to give us the total water potential here, which is just psi by itself. And in this case, we have a cell that's basically limp, it's flaccid, it's not turgid, and it has a pressure potential of 0, reflecting that. Its solute potential is negative 0.7. So the solute potential of pure water is 0. And so when we talk about solute potentials, they're always 0 in the case of pure water, or less than 0, because we have dissolved substances in the water that are binding the water and making it less free. Water can't be freer than it is when it's pure. And so here, the additive water potential then is negative 0.7 megapascals. So in this case on the left, we immerse the cell in a sucrose solution that has a solute potential of negative 0.9. All right, so that's a lower water potential than the water potential of the inside of the cell. And so water is going to be moving out of the cell more readily than moving in. And we're going to have plasmolizing of the cell. You see the cell membranes pulled away from the cell wall, and that cell's in big trouble. On the right side, we immerse the cell in pure water, which has a water potential of 0, solute potential of 0, and pressure potential of 0. This is just an open container, not under pressure in both cases. And you can see then the water potential is higher to the outside than to the inside, so water's going to be freer to move from the outside in than it is from the inside out. And we're going to have the water potential now inside the cell. The solute concentration remains the same, but the pressure potential increases to match that as the cell wall pushes against expansion of that cell, and it equilibrates at water potential of 0. And so this cell is now a turgid cell. So that's basically the concept of water potential. And we're going to think about that again now in terms of looking at transport throughout the whole plant at the level of the entire plant. And this is it can involve some major distances and huge amounts of water in the case of large trees like a sequoia dendron or a giant sequoia or a coast redwood, which can reach roughly 370 feet tall. So huge amount of water moving up through these trees in the course of a day. And as I mentioned, just in an average sized tree, we can have on the order of a ton of water being moved through the plant without any energy expenditure. And so the question is, how does this happen? So one possibility would be that the plant actually pushes the water from the roots up. And that sort of movement actually is possible in plants. And it happens most frequently at nighttime when the plant has its stomates closed. And the plant reaches its full hydration. And then we actually have active movement of minerals into the xylem. So through active transport, the plant has to concentrate some minerals in the xylem to move through the plant that are needed at a higher concentration than in the ambient soil solution. And by doing that, it creates a higher solute concentration inside the xylem than the solute concentration to the outside of the xylem. That is, there's a lower water potential inside the xylem than to the outside of the xylem in the cortex, and water will actually move into the xylem and place the contents of the xylem under positive pressure. And that can actually result in movement of water a few meters in a plant. And if you've seen leaves that look like this in the morning, prior to much or any photosynthesis occurring, this isn't dew that's condensed around the margins of the leaves or at the tips of grass leaves in many cases. It's actually water that's been exuded out of the leaves under positive pressure. And we call this guttation, this process of exuding water under positive pressure. But once the stomates open in the light and we have movement of evaporation of water from the inside of the leaves to the outside into the atmosphere, a process called transpiration, once that starts, this process of root pressure is completely overwhelmed by the pull from the leaves. So basically what happens is that we already mentioned most of the water loss from a plant is from the leaves and through these stomata, these pores that allow for CO2 to get into the leaves, which are needed for photosynthesis. Inevitably, that also results in water vapor leaving the leaves, because under normal atmospheric conditions, the concentration of water in the atmosphere is much, much lower than the saturated situation in the inside of the leaf, in the air spaces of the leaf, and water is going to be moving out. And the plant really can control that by controlling the opening of the stomata, but only to a certain point, and we're going to get water loss. And that water loss is going to result in the cell walls of the mesophyll cells starting to dry out. And a surface tension will develop on those cell walls that's carried into the xylary elements, the tracheids and vessel elements. And it creates basically the cohesion of water molecules, as you're aware, is very strong. And the tension of the evaporation of water from these cells